Hello mga scientistas! Welcome back to another episode of It's Science Time. This is your resident scientist, Andrea of 324 Bisaya Biology. For our today's segment, what I want you to do is to sit back, relax, and listen closely because we will have a hell of a ride for today's activities. What would be these activities? Well, first, we will know how to sterilize laboratory apparatus. Second, how to prepare culture media. And third, our last, is how to do serial dilutions. Despite the quarantine, we still need to know how to do these things inside the laboratory because we are scientistas. So, come on! In a laboratory setup, an autoclave is the proper equipment for sterilization. If the laboratory apparatus are soiled, we must clean the apparatus first before sterilizing because the sterilization will take minimal effect if the apparatus are soiled. The steam vent in an autoclave automatically closes when the pressure in the sterilization process is high enough to create a seal from the outside environment. This is why it is called as autoclave, which means automatic closing. This process of steam sterilization is crucial as it creates an optimal condition for the apparatus to be sterilized under the optimum temperature, humidity, and pressure. The usual pressure maintained in an autoclave is 15 pounds per square inch and a temperature of at least 121 degrees Celsius for 30 minutes to 1 hour. For our culture media preparation, we need to wear our proper PPE. Culture media contains In this nutrient agar that we created, microbes, specifically bacteria and fungi, will be cultured in this kind of medium. Close the containers. Once the mixture has cooled down, store the media invertedly. We invert Petri plates during storage or incubation in order to lessen the risk of contamination from airborne particles that may land into the agar. Get sample of microbes. When we are using plating method, different types are appropriate to what we want to identify. If you want to isolate single bacterial colonies, we will use strict plate method because of its method of cleaning the bacteria into individual cells, creating spaces in between. If you want to know the concentration of bacteria per ml, then pour plating is best used because of its method of pouring large amount of sample into the agar. If you want to enumerate viable bacterial colonies, we will use spread plating, as it isolates more specific bacteria because there is no subsurface bacteria that appears in this method. Soft agar overlay is used when working with PAGI to enumerate plagues. For our serial dilution, we still need to put our PPE on. Serial dilution is used to reduce a dense culture of cells to a more usable and countable number of separate colonies. For the materials, we need 10 containers with half water, soy sauce as our substance to be diluted, and a tablespoon. Using the tablespoon, we will pour the 2 tablespoons of soy sauce into container 1. Next is to get 1 tablespoon from container 1 and put it into container 2. This sequence will be followed into the next containers until it reaches the last container. For the final output, we can see the transition between the dilutions that we have made, and in cup 5, there is already little to no concentration of the soy sauce that we used because of the water or the transparency that the container 5 has. The number of colony forming unit is a factor in how many dilutions are needed for the sample. With the factor sample, individual tube, plating, and total series dilution factors combined can determine on the specific number of dilution. For today's recap, we have learned how to do sterilization using steam on our laboratory apparatus. Second, we have also discussed on the culture media or the preparation of this in order to isolate or to identify our bacteria in the sample that we have used. And lastly, we have learned about the serial dilution or how to control the number of the colonies forming units of the bacteria that we have in order for them to be used in a specific test methods that we are going to do in the laboratory. That's all for today, Scientistas. See you again next time!